Joining me now is Professor Zoltan Ivics, who is the head of the Medical Biotechnology Division at the Paul-Ehrlich Institute. Thank you for taking the time to talk with us today, Professor Ivics.
My pleasure.
Well, first, for all viewers who might not be familiar with the Paul-Ehrlich Institute, could you perhaps say a few words about its activities and expertise?
Yes, of course. The Paul-Ehrlich Institute is a very interesting institution with a double mission, where first of all, this is an institute that is a governmental institution in Germany. The double mission really means that it has a mixed area of duty. First of all, it's a regulatory agency in Germany. What does this mean? What it means is that it is the Paul-Ehrlich Institute that approves clinical trials and clinical applications of novel medicinal procedures and medicinal products for Germany, and also works together with the European Medicines Agency in the way of approving and marketing novel therapeutics and novel drugs and releasing them on the European market. So it's a complex area of activity that is related to securing the regulatory requirements, meaning certain safety aspects as well as efficacies of novel therapeutics in Germany as well as in Europe. That's kind of one of the mission of the Paul-Ehrlich Institute, and this mission is strengthened by a research activity. That is, the Paul-Ehrlich Institute is also a research institute with the vision of a synergism between the regulatory activities as well as cutting-edge novel experimental data, as well as novel methods, novel insight into biomedical research that actually speak together these two areas and strengthen one another in order to be on the top with respect to releasing novel therapeutics to the market.
Thank you. Well, one focus area of your work is called sleeping beauty transposition. Could you maybe briefly outline what a transposon is and what is special about the sleeping beauty transposon?
Yes, of course. And if now we could go to the slide just to highlight how a transposon works in nature. Let's just use very common, very simple language. A transposon is a jumping gene, a jumping genetic element that can change its position in the chromosomes. What is highlighted here on this slide on the left-hand side of this figure is a transposon, this particular jumping gene that is required, that requires one particular enzyme, a protein factor, which is called the transposase. These are the orange colored little dots that can bind to that can interact with the transposon sequence in the chromosome, and then through this, the transposon or this jumping genetic element is removed from its original chromosomal location, and then it translocates or transposes, this is the appropriate term that we're using, transposes over to a new chromosome, which is highlighted on the right-hand side of this figure. That is transposition. This particular genetic element, a gene, is jumping over from one location to another. And now if you click once more on that particular presentation slide, it already introduces the audience to the idea and the concept, how we can use the natural ability of these jumping genes to change their locations in the genome of a cell, and how we apply them as a research tool. And this is highlighted on the bottom of this particular slide, how we actually manufacture a gene vector system out of this transposon or transposition reaction. Namely, the the mechanism through which a jumping gene can insert into a chromosome can be turned into a gene vector. Now, what this really means in practical terms is that we take a piece of DNA, be it a fluorescent marker gene, a therapeutic protein encoding gene, a regulatory piece of DNA, practically speaking, anything. And we can clone this by using molecular biology technologies in a laboratory into a transposon vector, certain DNA sequences that we can maintain on a plasmid sequence. This is a circular piece of DNA that is also highlighted on the left-hand side. And now we generated an artificial transposon, because this is a transposable element that has now a DNA of interest or a gene of interest in a vector system. And we can conditionally express this factor, this protein factor, which is called the transposase. And once these two components of the transposition reaction are present simultaneously in a cell at the same time, the transposon DNA in form of the plasmid DNA and a genetically engineered plasmid and the protein factor, we can now catalyze this transposition reaction so that this gene of interest would integrate out of this plasmid DNA, out of the genetic vector, into the chromosomes of the recipient cell. So this is a very simple gene insertion technology through which we can provide plasmid DNA, and we can introduce plasmid DNA into cells, simultaneously provide a protein factor. And what will happen at the end of this reaction is that our gene of interest or our gene vector will be integrated into the host cell's genome and will permanently be inserted there and express that particular gene for longer periods of time or even lifelong. That's in a nutshell how transposition works and how we derive a certain genetic engineering tool vector system out of a naturally occurring transposable element.
Thank you. And this mini circle DNA at the bottom of the slide, I've read that this can be used for ex vivo, for in vivo gene therapy, but also for ex vivo modification of chimeric antigen receptors. Could you explain a bit how this can work?
Absolutely. We can just proceed to the next slide that kind of schematically summarizes how ex vivo gene therapy is executed in a nutshell by using transposon vectors. So you introduced already in your first question this name, Sleeping Beauty, and I like to kind of draw the attention now that this particular mechanism that I just described one slide ago is, of course, relevant not only to this particular transposon, Sleeping Beauty, but can also be applicable to a couple of other transposon systems. But perhaps the transposon system that is the most advanced in the context of a therapeutic setup is this particular transposon, which is called Sleeping Beauty, which follows that reaction that I just showed one slide ago. Now, in practical terms, how do we apply ex vivo gene therapy? Now, first of all, ex vivo means that cells that are recovered from a patient, isolated from a patient, are taken into culture. These cells are kept either in a petri dish or in a flask. That's why we use the terminology ex vivo. It's outside of the living organism or the body. And then in the context of a Sleeping Beauty transposon, what we typically do is to introduce ex vivo. So again, these cells are cultured in a dish or in a flask, and we introduce two factors that are relevant for this gene transfer technology. Again, a plasmid DNA or a mini circle, as you already mentioned, and this is something that is an advanced technology for providing transposon DNA, a kind of a mini circular gene that is devoid of antibiotic resistant genes, kind of a mini plasmid that is the most advanced form of supplying a Sleeping Beauty transposon. But anyway, the transposon carries a gene of interest. In the context of a gene therapy application, it's a therapeutic transgene cassette. This is component number one. Component number two is the transposase. What we typically do is either supply the transposase in the form of another plasmid or another minicircle DNA, or as this particular slide highlights, we can also in vitro transcribe the messenger RNA that is encoding the sleeping beauty transposase. This is important for certain safety aspects of the therapy. We mix these two components together, so straight naked nucleic acid components, and we typically transfect them into a cell population. For example, T cell population, because you asked the question of CAR T applications, so T lymphocytes, human T lymphocytes being the targets of this gene transfer experiment. This transfection step, at least in the current technologies, is typically done by using an electroporator technology or a nucleofection technology through which we physically transfer these two components into this cell population ex vivo. Following this electroporation or primary DNA transfer or nucleic acid transfer step, we typically culture these cells in ex vivo again in cells to allow for the cells to recover a little bit. We can also enrich the genetically modified cells, and we can even purify them away from the total cell population. And that what we typically do, as is also something that we do here at the Paul-Ehrlich Institute, is preclinical experimentation with respect to seeing how well, how efficiently, and how safely this entire procedure works. What preclinical really means is that we're running some either cell-based assays or running in vivo animal-based experiments to actually validate this approach. And of course, those colleagues out there who are interested in developing this procedure further into clinical applications would obviously end the ex vivo gene therapy cycle following a full documentation of the cell-based products by re-infusing these cell-based products into the same patient from which we originally isolated the cells. And this is how we complete a full circle of ex vivo gene therapy with a transposon vector technology, being quite likely CAR T applications, the most prominent examples for using this technology for clinical benefit.
Now, you mentioned the aspect of safety earlier. Can you tell me if there are any known advantages of the Sleeping Beauty transposon method compared to other techniques for T-cell modification, such as viral vectors?
Yes. Well, first of all, there are practical differences. Viral vector production, as well as working with these viral vectors, requires an S2 laboratory environment. This, of course, it's fully doable. Gene therapy has been done with viral vectors for longer periods of time, but it certainly represents a cumbersome step in vector production as well as manufacturing the cell-based products. Certain quality control assays are also relevant. I just named one particular assay which is certainly required for viral-based vectors, which is a replication competent, or the presence, or basically excluding the presence of replication competent viruses in cell-based products. This is all not required at all with a transposon-based vector system, because remember, we're talking about naked nucleic acid vectors. This typically requires an S1 laboratory environment, either straight plasmids or in vitro transcribed messenger RNA, or these minicircles require very simple manufacturing steps with respect to working with these vectors, working with the genetically engineered cells, and later on releasing genetically engineered cells for therapy. It's kind of an easier step, more straightforward manufacturing pipeline for a non-viral vector system. That's number one. With respect to safety, another important aspect is what happens with the genetically engineered cells. Obviously, the safety of the patient following gene therapy is of paramount importance. We have to make certain during approval of clinical trials that no harm is introduced into the patient. For example, a certain risk that could be linked to the actual vector system that we're using for genetically engineering a cell-based product. Now, transposons, typically the Sleeping Beauty transposon system has a peculiar feature of displaying a relative safety as opposed to integrating viral systems. And what I mean by that is that the insertional profile of the transposon of Sleeping Beauty is fundamentally different from the insertion profile of currently applied integrating vector systems, including gamma retroviral and lentiviral vector systems. Namely, as opposed to these viruses, Sleeping Beauty transposition doesn't prefer an expressed gene or a promoter region of an expressed gene for insertion. It's still not fully safe. We cannot target this transposon to a certain site in the genome yet. This is one particular project that we're currently running. But when we're comparing the overall genome-wide insertional pattern of Sleeping Beauty with viruses, it certainly displays advantages for safety. Namely, there's no preferential integration into genes. That also means there's no preferential integration into tumor suppressor genes and oncogenes, which is translated into an elevated safety of a gene therapy application over viral vectors.
That sounds promising. And what are the current challenges in the field and the obstacles that are left to overcome?
One of the obstacles, if we just talk about the core T field, but this discussion is probably also relevant for some other areas of genetic engineering or using genetically engineered cells for gene therapy to cure diseases. One challenge is to really bypass ex vivo gene therapy and try to introduce and administer gene therapy vectors directly in the body, which is an in vivo application. Now, obviously, the advantages of some of the viral vector systems is that by a simple intravenous injection or in vivo administration, they have a very powerful ability to transduce or genetically modify cells directly in the body. Now, the advantage of the transposon systems, obviously, as we have been just discussing, is that it's fully nucleic acid-based non-viral vector system. But on the other hand, this is also kind of a difficulty with respect of the system for in vivo applications. What current challenges include to really put together technologies, very efficient and safe technologies that could be used for coupling the nucleic acid-based vector system, just like Sleeping Beauty transposon. For example, certain lipoprotein complexes that the field is currently exploring through the application of which these non-viral vector systems could be efficiently introduced into target cells also in vivo. So this is a challenge and a very exciting area of research and development for the future. The other challenge that I just mentioned when we were discussing the safety aspects of this particular technology is, despite the fact that Sleeping Beauty transposition is already safer with respect to genomic insertion preferences than many of the or some of the viral vector systems, one challenge that is still remaining is to put together technologies through which we could put our transposon integration into any site of the genome at will. Practically speaking, what this would mean that we would guide the integrating transgene copy into a certain locus or to a certain site in the human genome through which we could enhance the safety aspects of the resulting cell-based product tremendously. there's very good indication that, and proof of concept indications that this can be done also with Sleeping Beauty vector systems, but we are not there yet to really release that technology. The efficiency of this technology is not at the level yet that would be required to be really advancing to a level of clinical application so that we could elevate Sleeping Beauty transposition safety-wise to the next level. So this is another challenge that we're facing. Otherwise, and maybe this would be still an opportunity to show one more slide in the context of CAR-T applications, if you could just click through quickly and then show kind of a summary, a summary thought with respect to that although these challenges are still there with Sleeping Beauty Transposon, the applications that we and the efficacy and the safety that is linked to Sleeping Beauty Transposon vectors in a clinical context, it certainly matches the current technologies based on viral vector systems in terms of efficiency. What this particular slide shows, that with these mini-circle technologies, we can reach roughly 50% of T lymphocytes by expressing a CD19 CAR in these lymphocytes by a simple electroporation technology. This is as good as it gets with any of the other viral vector systems. The bottom panel of this slide actually shows that by Sleeping Beauty engineered human CAR T cells, you can eliminate a CD19 positive human lymphoma in a xenograft mouse model. So biological activity and efficacy is clearly there. That's number two. And number three, which we already discussed, the safety aspects clearly provide an edge and an advantage to this Sleeping Beauty technology over the viruses. So despite the challenges and the next phase of preclinical development to elevate this technology for in vivo application, as well as elevate this technology and further advancing its safety aspects, it is already there to clearly match, if not exceed, safety as well as efficacy of other engineering or genetic engineering tools, including some of the viruses, and with the added value of working with a straight nucleic acid molecules that provide simplicity, relative safety, and a relatively cheap GMP manufacturing of these vectors for clinical applications.
So you mentioned that for clinical applications in the long run, it is still quite a long way to go. But now I'm curious, how do you imagine, how could this look like in the future, the translation of this research to clinical practice and industry application?
Well, maybe I use the opportunity to just highlight a multi-center clinical trial that has been funded by the European Commission through the Horizon 2020 Research and Innovation Program. This consortium is a pan-European, multi-center European consortium aiming at clinical translation of Sleeping Beauty technology in the context of multiple myeloma by using CAR-T engineered T lymphocytes. I've been fortunate to be a partner in this consortium and contributing with research activities. For example, this particular technology based on mini-circle vectors and the entire Sleeping Beauty technology for gene delivery, as well as addressing the safety aspects of Sleeping Beauty engineering of human cells. And this particular clinical trial has 4 clinical centers, one in Germany, one in Spain, one in Italy, and one in France. And I'm happy to report that 2 sites, the German and the Spanish site, has been cleared, has been given the green light with the respective regulatory agencies for starting their and recruiting their patients for clinical applications in this particular clinical trial. So this is obviously a very exciting area because I have been working with the Sleeping Beauty Transposon in the past 20 years. So it's a great opportunity for me to contribute with my research activities to these applications. And if I can project these clinical applications, or what I see and what I witness today, certainly my hope and my projection would be that quite likely this technology will be picked up by developers, academic developers, as well as clinical developers and companies alike, quite likely on the basis of the relative efficiency, safety, as well as economical advantages this particular very simple DNA-based technology can represent for CAR-T applications, but also for other applications of gene therapy, including hematopoietic stem cell engineering or engineering other types of cells. So I am very hopeful and very optimistic with respect to kind of pioneering through the first clinical applications, not only here in Europe, but also in the United States, namely CAR-T applications. This area will be picked up and will be explored even more heavily by developers on the basis of these particular advantages of this genetic engineering technology.
That's very interesting to hear. Thank you very much, Professor Ivics, for these insights. And thank you very much for joining us today.
My pleasure. Thank you very much too.